Carbohydrates form a class of biomolecules apart from proteins, nucleic acids and lipids. The roles which the carbohydrates play are so diverse in the entire life processes, from starting from the simplest bacteria to human beings. It is a point of wonder that they are structurally so different and functionally so different, and so many types of carbohydrates are present. So let us understand what carbohydrates are. The simple definition is the carbohydrates are aldehyde or ketone compounds with multiple hydroxyl groups, OH groups, and having the empirical formula of CH2O whole N, and N stands for the number of carbon atoms. They form one of the four major classes of biomolecules along with proteins, nucleic acids and lipids, and they play extensive roles in the life processes. The major roles they play are they serve as energy stores, fuels and intermediates of metabolic pathways, several metabolic pathways, and they are also form the structural framework of RNA and DNA. We all know that ribose and deoxyribose forms a part of the RNA and DNA molecules, and deoxyribose and ribose are all carbohydrates. Now comes polysaccharides. Polysaccharides are all derived from monosaccharides and they are actually carbohydrates. Polysaccharides are structural elements in the plant cell wall and in the bacterial cell wall. Also, some polysaccharides serve as energy resources and storage of energy in the form of glucose polymers in starch and glycogen. And carbohydrates also are linked with proteins and lipids in some cases, and they form glycoproteins and glycolipids, and these glycoproteins are present on the surface of cell membrane, and they are pivotal in maintaining the cell-cell communication. That means they mediate in cell signaling. Wherever there is an external stimulus, the signal has to be transmitted into the cell and via these glycoproteins and glycolipids in some cases where they are present on the surface of the cell membrane. So the building blocks of carbohydrates are monosaccharides. The simplest and the smallest monosaccharide is dihydroxyacetone and D and L-glyceraldehyde. In this case, the number of carbon atoms, that is N, as suggested in the empirical formula, N is 3. So the monosaccharide is a triose. Tri means 3. There is a difference between dihydroxyacetone and glyceraldehyde, whatever D or L form, because in both the cases N is 3, but what is the difference between them? Difference is dihydroxyacetone has a ketose group. It is a ketose sugar, keto group, C double bonded O, and glyceraldehyde is an aldose sugar because it has an aldehyde group. It is evident from the structure shown. Now comes the difference between D and L-glyceraldehyde. What is the difference between D and L-glyceraldehyde? D stands for the asymmetric carbon atom which is present in the glyceraldehyde molecule, and with it the hydroxyl group is attached. In the D form, the hydroxyl group is attached is in the right hand side, and in the L form it will be in the left hand side. But no such problem arises in case of dihydroxyacetone because it does not have any asymmetric carbon atom. And by definition, asymmetric carbon atom is the carbon atom which has 4 different groups attached to it to maintain the tetravalent form structure. So in case of D-glyceraldehyde, there is one such asymmetric carbon atom. And other monosaccharides having number of carbon atoms as N is equal to 4, 5, 6 or 7. The monosaccharides are tetroses, pentoses, hexoses, heptoses respectively. And the number of asymmetric carbon atoms obviously gets increased in case of tetroses, pentoses, hexoses and heptoses. Presence of multiple asymmetric carbon atom creates the formation of enantiomers and diastereomers. And the capital D and the capital L designate the absolute configuration of the asymmetric carbon atom which is farthest from the aldehyde or the keto group. And that carbon atom, that asymmetric carbon atom is considered for designating the D or the L configuration. So theoretically, the number of possible stereoisomers equals to 2 to the power n, where n is the number of asymmetric carbon atoms. This n is different from that empirical formula n. So this n stands for the number of asymmetric carbon atoms, and in a 6 carbon aldose such as glucose, which has 4 asymmetric carbon atoms, there can exist 16 all possible stereoisomers, and glucose is one such isomer, one member out of the 16. Next slide shows the formulae of the D-aldoses. We have started from D-glyceraldehyde, the simplest aldose, and we have ended in the hexose portion, where the number of carbon atoms is 6. And all these cases, it has been shown that the D isomer exists freely in nature, not the L. Isomers are chemical compounds having the same molecular formula but different structures. So isomers can be classified as constitutional isomers and stereoisomers. And constitutional isomers differ in the order of attachment of the atoms. We can see that glyceraldehyde and dihydroxyacetone are constitutional isomers, though the molecular formula in both the cases are same, but one has a aldehyde group, one has a keto group. That means they are constitutionally different. And in case of stereoisomers, the atoms are connected in a same order but in different spatial arrangements. So stereoisomers can be again classified as enantiomers and diastereomers. What are enantiomers? Enantiomers are the non-superimposable mirror image compounds. So D-glyceraldehyde and L-glyceraldehyde are enantiomers because one is the mirror image of the other and one cannot be superimposed on the other. Very simple example is the two hands. One cannot be superimposed on the other and one is a mirror image of the other. In diastereoisomers, these isomers are not mirror images at all. So diastereomers are again classified as epimers and anomers. Epimer, what are epimers? Epimers differ at one of the several asymmetric carbon atom. So D-glucose and D-mannose are epimers. Again, anomers are separate class of diastereomers where molecular formula has been depicted in a ring structure. Actually, in nature, the ring structure exists, not the straight chain structure. For the formation of an anomer, the ring closure has to be taken place, and this creates another asymmetric carbon atom. So, after the formation of an anomer, a new asymmetric carbon atom is created, and this gives rise to different types of isomers. So, in case of alpha anomer and the beta anomers, anomers are designated as alpha and beta according to the orientation of the OH group which is attached at the newly created asymmetric carbon atom as a result of ring closure. In this slide, it shows how the ring has been formed. Ring closure is possible involving, in case of glucose, the carbon number 1 is involved with carbon number 5 to form the ring structure. And this forms a 6-membered ring, pyranose ring. The word pyranose comes from the compound pyran. It has similarity like the pyran. So, glucose, D-glucose open chain form has been converted into a closed ring form forming alpha D-glucopyranose, and this alpha suggests that it is an alpha anomer. The beta anomer and alpha anomer can be differentiated by the orientation of the OH group which is newly formed as the newly synthesized asymmetric carbon atom. Again, D-fructose. Fructose, it is present in fruits, the fruit sugar. Fructose, there is another ring formation takes place, and the carbon atoms involved is carbon 2 and carbon 5, and the ring form is called alpha D fructofuranose. This ring is a 5-membered ring called furanose ring. This resembles the furan compound. And the 6-membered rings are spatially stable, but 5-membered furanose rings are also stable and they also exist in carbohydrates. In equilibrium mixture, the ring forms of most sugars predominate over the open chain forms. Simplest D-ketose is erythrulose. The common ones are ribulose, xylulose, where number of carbon atom is 5. Then fructose is the most important, ketohexose, and then pseudoheptulose. So the typical sugar with the widest distribution in nature is glucose, D-glucose. Actually D-glucose, the D isomeric form, and in this case the ring formation has taken place from the straight chain form, and the pyranose form ring forms the hemiacetal formation has been possible. This hemiacetal formation creates the pyranose ring. So the projection formulae, which is depicted in the textbooks or whatever in the printed material, these follow the Fischer convention, and in the Fischer convention the ring has been depicted in this way. Alpha D glucopyranose suggests that the alpha anomer is considered, and the glucose monosaccharide has formed a ring forming a pyranose structure, that is a 6-membered ring is formed. But in case of beta D fructofuranose, this suggests that the beta anomer is considered, and fructose monosaccharide has formed a 5-membered furan ring, furanose, hence the name. According to the Fischer convention, each carbon atom must be viewed with both vertical bonds projecting behind the atom which is viewed. But Hayward structural formulae are nowadays used, that the heavy lines which are drawn, these are thought as projecting out towards the viewer, and the other edge is projecting behind the plane of the screen. So the ring structure molecule is not planar. Then what is the actual structure of the pyranose and the furanose rings? These rings assume two different types of conformations, and these rings are not planar, as I have already mentioned, and the pyranose rings can adopt two types of conformations, the chair form and the boat form. The names suggest that the structure resembles the chair, and that the boat form structure resembles the boat. And the groups attached to the ring carbon atoms have two orientations, the axial and the equatorial. In the axial orientation, the axial bonds are nearly perpendicular to the average plane of the ring. More or less, the axial bonds are perpendicular to the average plane of the ring. And in the equatorial orientation, the equatorial bonds are nearly parallel to the plane, average plane of the molecule. Steric hindrance means if the R groups or the constituents which are attached to the ring carbon atom are quite bulky, then there will be a steric repulsion between the two adjacent groups, and this causes steric hindrance, and steric hindrance suggests the reason behind the destabilization of a structure. So it is not favored. So normal conformation will be such that there is minimum steric hindrance, and equatorial substituents are less crowded, but rather than axial substituents are. Let us have a look what are the types of alterations which can be done with monosaccharides after they participate in a chemical reaction, like joining with alcohols, joining with amines via the glycosidic bonds. The monosaccharides are linked to form complex carbohydrates, because monosaccharides are the simplest forms, and as we know that carbohydrates has so many diverse structural forms, so monosaccharides has to undergo transformation forming carbohydrates, complex carbohydrates, and they are linked with each other by the oxygen O glycosidic bonds. O stands for oxygen. So the glycosidic bond has been formed involving an oxygen atom, and a few monosaccharides are linked to form an oligosaccharide. As for example, the disaccharide maltose has two glucose residues joined by the alpha 1,4 glycosidic bonds. So alpha means the alpha anomer has been taken, and 1,4 means suggests that one carbon atom of 1 and carbon atom of 4 of second glucose has been joined with the removal of one molecule of water. Oligosaccharides have a polarity defined by their reducing and non-reducing activity as they form open chains. From where comes the reducing property? The reducing property comes from the highly reactive aldehyde or keto groups of the monosaccharides. The polysaccharides are, poly means many, and polysaccharides consists of many monosaccharide units joined by either alpha or beta glycosidic bonds. So these are the few which are very common and all of you must be acquainted with: sucrose, maltose, lactose. In sucrose, the glucose molecule and the component and the fructose component are joined by alpha 1,2 linkage. In case of maltose, alpha 1,4 linkage has been participating to join two glucose residues, and in case of lactose, beta galactose and alpha D glucose has been joined. And all these three disaccharides are extremely important. Other modifications of carbohydrates may be possible at different positions. This slide suggests which are the potential sites or positions which can be modified or which get modified during subsequent chemical reactions, and these modified carbohydrates play highly basic and highly specific functions, so their modification is extremely important and extremely necessary for the maintenance of the life processes or the living cells. So this carbon 6 has the CH2OH group, and this can be modified with a COO minus group, acidic group, and it forms glucuronic acid. It forms an acid. So, this glucuronic acid is a component of mucopolysaccharides. Then CH2OH group has been replaced by a sulfate molecule. Then again in several places OH has been replaced by H. So this is anhydro form. Several modifications are possible and they are actually done in the nature and form some modified carbohydrates such as muramic acid, which is a component of the bacterial cell wall. Then comes N-acetyl neuraminic acid, which is also called sialic acid, and it is present on the surface of cell membranes in eukaryotic cells. So the list is very long, so not all the modified base positions are cannot be discussed in such a small time. One is N-acetyl neuraminic acid, is very important for maintaining the bacterial cell wall, and N-acetyl neuraminic acid is important for maintaining cellular glycoprotein structures of the cell membrane. Then comes 2-deoxyribose, beta configuration, that is beta anomer has been considered, and 2-deoxyribose is famous because it forms a structural framework of DNA. L-rhamnose, L-fucose, these are all present on bacterial cell surface. Now comes polysaccharides. So what are polysaccharides? Poly means many, and these polysaccharides are formed by joining many monosaccharide subunits. So monosaccharides are the simplest structural subunit or the moiety which comprises of the polysaccharide, and polysaccharides are classified as homopolymers and heteropolymers. Characteristic reactions are, this means that the aldehyde group or the keto group, these are highly reactive. They can undergo both oxidation and reduction. Aldehyde groups can be oxidized to form acid, or on the other hand it can be reduced to form alcohol. So oxidation of D-glucose by alkaline copper sulfate, that is Fehling solution. Fehling solution you must be knowing. Fehling solution has alkaline copper in the copper divalent copper, and it gets reduced to Q plus oxide monovalent copper, so reduction, and in the meantime the carboaldehyde group has been converted or oxidized into the acid group. Similarly, the aldehyde group can undergo reduction by using sodium borohydride, and the formation is the CH2OH group, that is sorbitol. The compound formed is sorbitol, which is formed after the reduction of D-glucose by sodium borohydride. Another unique reaction is the periodate oxidation. Here the carbon-carbon bond is cleaved. Which carbon-carbon bond? The carbon-carbon bond where the carbon atoms has adjacent OH groups with it. So in case of monosaccharides, this is possible. Adjacent carbon atoms have OH groups, and that carbon-carbon bond is cleaved and by periodic acid or sodium periodate, and this is called periodate oxidation. It is also called Smith's degradation because the carbohydrate will get degraded. I have already mentioned that glycosidic bonds are formed, and this is the mechanism how the glycosidic bond is formed. In case of glucopyranose, alpha D glucopyranose, two molecules are joined with the elimination of water, one molecule of water, and the formation of a maltose or the disaccharide maltose, and the bond is called alpha 1,4 glycosidic bond, and that means alpha glucopyranose is the anomer which is involved in the reaction, and carbon 1 of 1 glucose and carbon 4 of 1 glucose is involved in this bond formation, and with the OH of 1 carbon and H of 1 carbon gives rise to water, and water is eliminated and a glycosidic bond is created. So in case of any other alcohol ROH, so this H and OH of one carbon 1 of the monosaccharide joins to form water, and water is eliminated and OR has been linked with the monosaccharide, and this portion is called aglycome. Other characteristic reactions of monosaccharides are number 1 is boiling with concentrated HCl and H2SO4 and the conversion of pentose sugars. It is a peculiarity for the pentose sugars only, and they are converted into furfural pentose sugars. Then resorcinol in 3 molar HCl gives a red precipitate with ketose sugar. This is also unique for in case of ketose sugars. Then diphenylamine with sulfuric acid, it gives a blue-green color specifically with 2-deoxy sugars, and therefore it is specific for identification of DNA because DNA has the 2-deoxy sugars. Then glucose forms osazone derivatives when reacted with phenylhydrazine. Phenylhydrazine is C6H5NH NH2, and this osazone which has been formed with glucose forms the compound is called glucosazone. Fructose also can form osazone with similar reagents. Then methylation. The free OH group of carbohydrates can be methylated by treating with methyl iodide. One thing, a characteristic reaction in case of disaccharides, only I will like to mention the characteristic reaction of sucrose. Sucrose undergoes inversion, that is inversion of sucrose. What is inversion? All these carbohydrates molecules which have a asymmetric carbon atom has the property of optical activity. Optical activity means they can deflect or rotate a plane polarized light either in the right hand direction or in the left hand direction because of the presence of the asymmetric carbon in the monosaccharides. So if the plane polarized light is rotated in the right hand side, then it is called the molecule which is capable of doing this rotation or undergoing this rotation is called a dextrorotatory molecule, and if it is in the rotation occurs in the left hand direction, then it is called a levorotatory. These monosaccharides are optically active, and sucrose also is optically active because of the presence of several asymmetric carbon atoms, and in nature sucrose can rotate in the plane polarized light in the right hand direction, and it is hence it is called dextrorotatory. But sucrose, as already has been shown, has a glucose molecule group and a fructose molecule. So sucrose undergoes hydrolysis in presence of mild acid and slight heating, forming glucose and fructose. And glucose in general normally is dextrorotatory, but sucrose is dextrorotatory, glucose is again dextrorotatory, but fructose is levorotatory. And the optical specific rotation of fructose is higher in the negative direction rather than in case of glucose. The rotation the value is around 54 degree plus 54 means it is dextrorotatory. Sucrose glucose is dextrorotatory, and fructose is the specific rotation of fructose is around 92 degree, minus 92 degree. Hence the net components will give a levorotatory component around minus 39 degree. What is mutarotation? Alpha and beta anomers can interconvert. Alpha anomer of glucose can interconvert into beta anomer, and in equilibrium there is 36% of alpha anomer and 64% of the beta anomer. So at equilibrium these two components collectively form or collectively contribute to the specific rotation of the entire glucose solution, glucose molecules. So beta anomer is also dextrorotatory and alpha anomer is also dextrorotatory, but the specific rotation of alpha anomer is plus 112 degree and specific rotation of beta anomer is plus 18.7 degree. But at equilibrium beta and alpha are present as 64 and 36% respectively. So at equilibrium the specific rotation of glucose is a mixture of both these anomers, and this is the value is around 52 or 53 degrees. From this lecture we want to focus on the idea that monosaccharides, which are very vital for the formation of polysaccharides and for the maintenance of the life process in the cell. So monosaccharides are the building blocks of a very versatile group of biomolecules which are essential in maintaining the cellular functions and other cellular activities such as cell-cell communication, energy release by the cell or synthesis of energy, metabolic energy, and so on.